With the comprehensive nature of genetics, we bring you a segment about epigenetics and genes expression. Our first speaker in this session is Dr. Mili Thakur. Dr. Mili Thakur, I have the honor to introduce her again. Last genes, I also introduced her. She is a medical doctor. She is triple board certified specialist in reproductive endocrinology, medical genetics, and obstetrics and gynecology. With over 2 decades of expertise, serving as a physician, scientist, entrepreneur, speaker, and content creator, Dr. Thakur founded Genome Ally, a telehealth-based reproductive genetic practice. She is specialized in proactive genetics, offering comprehensive consultation and testing for individuals on a family-building journey. Dr. Thakur's expertise is focused on prevention of genetic diseases in the offspring through in vitro fertilization and pre-implantation genetic testing. Additionally, she serves as a reproductive endocrinologist at the Fertility Center in Grand Rapids, Michigan. An award-winning scientist and clinical assistant professor at Michigan State University, Dr. Thakur is dedicated to advancing medical research and education. She has contributed to the FACOG Clinical Consensus Gynecology Committee and the SART International Committee, exemplifying her commitment to shaping clinical standards. Please welcome Dr. Mili Thakur.
Hello, everyone. This is an exciting upcoming topic. I think researchers in the field have amassed so much data in the field of epigenetics now that it's going to take off. And this is an exciting time in our field. So first of all, I wanted to thank the organizers of the meeting. Thank you, Dr. Raj and Fernanda. And thank you to all of the participants and experts who are here. Because the meeting is such a scale, like I could talk to most everybody and learn a lot. So I enjoyed the meeting. Thank you. So these are my disclosures. I am a founder of Genome Ally, a telehealth reproductive genetic consultation practice. I'm also a speaker and advisor for Natera, which is a genetic testing company. I am a persistent optimist. So a lot of the things that I say would feel a little flowery. But the reason why I do that is because there are researchers within you who would take that idea back and then make it happen. So I do also endorse client-informed decision-making and pre- and post-test genetic counseling. So a lot of things that I present, they're heavy on that pre- and post-genetic counseling aspect of it. So the objectives of my talk is to discuss the variability. Is it working? Yeah, so I'll be discussing the variability in IVF success. We saw a lot of talks from yesterday where we saw that there is variation in the success of IVF, introduce the concept of epigenetic programming, discuss how we can learn from big data technology to enable personalized approach to fertility care and IVF, and now introduce also the concept of how AI can help with that management of that big data, and then gain insights into leveraging epigenetics to refine and enhance contemporary infertility care practices. So this slide basically shows you the decades that we've had. We've had 40 years, and this is the average number of intended retrieval per live birth and delivery. So if you see, for somebody aged less than 35, Despite the advancements we have made, you would need an average of 2 cycles to take a baby home. And then between age 35 to 37, about average of 2.8 cycles. Between 38 and 40, 4.3 cycles. And then above the age of 40, 12.7 cycles would be required. And this is despite decades of innovation in the field. Our success rate is not great. Almost less than half of the time, the baby will go home. And IVF success can vary significantly across different practices and among patients at the same practice. Who here has seen IVF cases between the same patient where the success or the number of eggs was like drastically different? All of us have, right? And usually when I talk to clinicians or people who are taking direct patient care, they would say, oh, those patients have differences with their age. We know that younger women will typically have higher success rate. The ovarian reserve matters a lot. So if your AMH level is great, the quality and quantity of eggs would vary, and how Dr. Cooper showed, there would be a difference in the number of eggs retrieved, which is a big number for us to focus on because that dictates how many blasts we are going to get, and the quality is going to dictate how many of those are going to be euploid, and that dictates the success. Then the health conditions matter, right? The patient with PCOS or endometriosis or fibroids or premature ovarian insufficiency are on different spectrums of success just because of those health conditions. And then there are technical and procedural differences. We all agree that not every lab is the same, right? The laboratory techniques are not same across the board. Embryo culture conditions can vary even if there is a slight fluctuation. And yesterday we heard talks from embryology experts about how a small change in what filter we are using in the lab or what kind of air is going in, there could be changes in the success. And of course, the transfer protocols. For all clinicians who work in this field, we agree that the most important procedure in the whole field of reproductive endocrinology is the embryo transfer. And if by chance on the day of your transfer, you touch the top of the fundus and there is a little bit of bleeding, everything else that you've done all across the board, the embryo is not gonna implant in that situation if there was bleeding with the transfer catheter. So we would say, oh, between different patients or within the clinic, these things could matter. If you have different physicians doing different transfers, their success rates could vary, right? And then the expertise and experience of the medical and embryology team can differ. And that's why there could be different successes. So what I mean by that is if your patient just didn't trigger at the right time and you had premature ovulation, that success rate is going to look very different than if you were able to retrieve all the eggs that they could have. However, within the same practice, using similar protocols, the same embryology personnel and lab environment, if they are kept same, there is still variable success among patients. So there are some patients who will come in at the same age, so less than age 35, they all have male factor, but the number of eggs retrieved between those patients can be different, their blast conversion rate can be different, and also their aneuploidy rate can be different. And I'm sure you've thought about this if you work in the field of reproductive endocrinology. It appears success is influenced by a multitude of factors beyond the control of clinicians and technology. So many a times, as a clinician, you really want a good blast to transfer, right? So you go to the embryology lab director, and one time I went as a new person coming out of fellowship. I really wanted my patient to have the success, and I went there. And they told me, you're going to get a blast of whatever you're going to give us with the eggs, right? The embryology lab is not able to enhance the quality of whatever is coming to them. And it feels like something else is at play. So within the practice, why would it be different? And within the same patient, why would the success be different between that same patient cycle last month, where we got 4 eggs, and this month, where we now got 8 eggs? It's the same protocols. Maybe there is a change in protocol. So what are we overlooking? What changes from cycle to cycle? Why is the aneuploidy rate in a certain cycle different than the next one? So the patient changes from cycle to cycle. It's not the same patient when you're going from one patient to another. Plus also the patient that you stimulated, the same patient who's now 34 years and 1 month older, is not the same patient that we treated the last month. That patient is changed from what she was, or that male partner who that person was the last month. So they have changed now. So what I mean by that is that the genetic and epigenetic factors, they modify and influence our gamete and embryo quality and implantation success. And there is variability in gene expression and epigenetic programming among the patients. So epigenetics is this science, which we will talk about, that influences the change of a person from one to another. So what I mean by that, all of you here, when you woke up this morning, are now slightly different from what you were a few hours ago. For those of you who had coffee this morning, how many of you had coffee this morning? So all of you had coffee this morning are different from what you slept last night. And then how many of you had alcohol last night? So all of you who had alcohol last night are slightly different than what you were. So as soon as that input came to your system, your body decided to do something different. A few of the genes were turned on and off, and we'll talk about it. So you're slightly different. And today, if you drank enough water, by this evening, you would be a different person altogether. So we always talk to our patients about big things, right? Smoking, cessation, alcohol consumption, and diet and stress level management. And we know that they change outcomes. And then environmental exposures and lifestyle choices will change the outcome. Now, why are we not talking about these things? And we'll kind of talk about it because the field of epigenetics is full of layers and layers and layers of information and data. It's really hard to study these effects. And in order for the audience to get on the same page, I'm going to just break it down. I know there are some researchers here who do research in epigenetics, so they're very well versed with the field, but there are others who are just getting to know the field, or like how I was a few years ago, just kind of scratching the surface of the field. So let's talk just basic genetics here. We all have billions of cells. All of those cells come from that first cell where the egg and the sperm came together. And all of that information is stored inside the nucleus of the cell in the form of chromosomes. We know we have 46 total chromosomes. There are 23 pairs. The first 22 pairs are the ones that are same in boys and girls. And then the last pair is what makes us a girl or a boy. If you have two X chromosomes, it's a girl. If it's an X and a Y, it's a boy. Each chromosome pair consists of contribution from both the maternal and the paternal origin. So each one of these chromosomes has to come from a mom and from the dad. If it doesn't, then we call it uniparental disomy, and that's an issue. So we are all familiar with this pathway, right? Basic biology. We have the DNA, which has been the traditional gatekeeper of all the information. It continues to be the gatekeeper of the information. This genomic DNA is what we study with our PGTA technology, right? And then the transcription happens where mRNAs are made. Then translation happens when the proteins are made, and those proteins are the ones that are going to be the biological building blocks of the information in our cells. So now what is epigenetics and why is it important? So epigenetics is basically the regulation guidebook for cellular behavior. So a good example I want to give you is, if what I said before was true, that means all of our cells in our body come from that first cell that has all of the 46 chromosomes, right? 23 from the mom and 23 from the dad. Then my brain cells and the red blood cells have the same genetic material, right? So they are at liberty of trying to do all the things that are coded in all of our 20 to 30,000 genes that we have. But they do different functions. So basically, epigenetics modification is the modification or the layer that is put on top of the genetic information. So brain cells can do cognitive processes, and then red blood cells can carry oxygen. So some genes are switched off in the red blood cells that are not making the right protein for cognitive process, but the ones in the brain are making proteins that are required for them. So early on in our development, these cells have genes that are switched on and off, and they stay most of that way, but then in certain conditions or with certain exposures, that can change over time. And this provides each cell type with a distinct functional specification. This process is essential. It happens for all of us in different ways. So a good analogy would be if these chromosomes are like books that are stored in the library of the cell. There are 23 pair of encyclopedias with book A and book B for all of them. And inside of those, there are chapters. There are different chapters which are the different genes. And based on like what epigenetic bookmark has been put onto that cell, then that cell will have some chapters that you can read. It determines which part of our genetic instructions are read and followed. Epigenetic controls where genes are turned on and off, similarly to like a light switch. And exposure to different environments will change that light switch and switch on a different light switch. And this regulatory process enables our bodies to adapt to different situations. This system is highly dynamic, and whether we are aware of it or not, it's always listening. So even if you might be a strong person and you can go through hours of intense activity, you trained as an athlete, or you can drink 10 cups of coffee and be really okay, your body is still listening to its own score. It's only listening to what it's been programmed to as an evolutionary memory or as a physiological memory. It doesn't care how gritty you are or what you have to pull through. It will do what your mind wants you to do, but then really on the cellular stage, everything is being tracked and kept without your permission. So these bookmarks are regulating these gene expressions, and whenever there is a gene activation that happens, the genetic code is read and the right protein is made based on that. When there is gene suppression, there is disregard of the instruction, and there is no actual change to the DNA sequence, but it determines which areas become accessible for transcription and then utilization for the body. And all of our environment is constantly interacting with our system. Again, I kind of bring it to your attention, it's not your permission. If you were exposed to a draft of cold air, something slightly changes in your body. You didn't want it to change, but it's changing no matter what you do. The same is happening for our patients. So a good example that I kind of use is many of our patients, like when they're sitting in the waiting room, they're scrolling social media. So the egg retrieval is going to happen in an hour, right? Or half an hour, we have to get them ready. They're scrolling social media, and their cortisol level is rising every time this woman sees a baby on the picture. So the eggs that I'm gonna get now versus an hour before she started scrolling on her social media are different. They have changed from what was prepared by the whole IVF team and the patient together over the course of 15 days. That exposure in the last hour or so is gonna change the quality of those eggs. And these environmental factors are constantly being given to all of us. So what are some of the mechanisms? The researchers in the field can give you more detail about it, but the short answer is one of the mechanisms is DNA methylation. There are these tiny tags, the methyl groups, that are added to the DNA. They selectively cover the certain genomic regions. Methylation offers, it inhibits the transcription and utilization of tagged DNA, and it's basically like a do not disturb sign. So Dr. Cooper talked about Fragile X a little bit ago, right? When there is full mutation for Fragile X, then the methylation silences the gene, the Fragile X gene, and no protein is made, and therefore Fragile X syndrome happens, and there is problem. But when there is pre-mutation, this methylation is not completed. So there is still transcription of the mRNA, and there is toxicity, and that's why they have premature ovarian insufficiency. So one of the mechanisms is methylation. The other mechanism is histone modification. So lengthening or shortening of the histone segment, is going to change. So histones, are they serve as spools around which DNA is wound. So if you look in the picture, our DNA is really long and it has to be stored in these chromosomes, so it's wound around the histones, different interaction with different environmental things. This tightening or loosening of the DNA coil changes, and that makes it, whether or not the gene instructions can be read. So in this, if it's tightly wound, the instructions, are not being read so well, and if it's loosened, then the instructions can be read. And then there's another mechanism called non-coding RNA. Most of the RNAs will code for a protein and make the protein in the body, but then there are these other RNAs that don't code for protein. Instead, they regulate gene function. And these are conveying and taking information in and out of the nucleus, and then they're regulating the portions of the genes that will be read or not read. So this has now, we have collected enough data now that has now led to the association of epigenetics with a lot of human disease. So epigenetics in obesity, type 2 diabetes, how does meditation or mindfulness practices change our epigenetics, exercise and epigenetics, your placenta and epigenetics, the imprinting programming of the next generation is through epigenetics, your epigenetics with different protein-based inheritances, and then the new era of epigenetics of epidemiology. So in essence, what I'm trying to say or convey the message to you is every single interaction that you're having with the outside environment, including your food, including your exercise, including whatever environmental exposures we are or not exposed to, is because of epigenetics. And there is studies that these researchers are coming out with that are associating memory going over into multiple generations. I'm not sure how many of you know about this evolutionary memory that certain ethnic groups will actually have, and that's passed on to their next offspring in humans and in other mammals, that it can be passed along. A lot of data coming through about aging and restoration of how we can reverse aging. For those of you who were here last year, I mentioned a couple of books, and one of them is David Sinclair's How We Age and Why We Don't Have to. If you haven't read it yet, it's a good read. There's a epigenetic perspective of our health span, how cancer works. All of this ties into that genetics of cancer, how different exposures work, right? And how we unlock, different disease treatments. So how are we taking care of our patients right now? So most of the providers, the clinical providers in the group, will do an excellent job of changing IVF stimulations based on different patients. So patients who have PCOS, we want to stimulate them with the lower dose. We want to trigger them a certain way so they don't develop ovarian hyperstimulation. We usually will do a freeze-all for them. We will cater, we will take some basic information from the patient. We will dose a patient with a higher BMI, higher for a trigger dose, right? And then we will also change how we dose the medications based on their age. And now with all the accumulated data over the last few years, we don't want to dose too high and push these follicles with high doses of FSH. So all of us are changing the protocols, but most or the majority of our IVF stimulation, trigger, and embryology protocols are same for every patient, right? For most of our patients, we are stripping the eggs the same way, we are doing ICSI the same way, we are culturing them the same way. And so is our stimulation and trigger. Most of us are using a combination of either gonadotropins, right, or some sort of oral medication and gonadotropins. We are triggering with just a few medications. So this current size of one-size-fits-all approach may not address individual patient needs effectively. And there is limited ability to tailor treatment to each patient's unique genetic and epigenetic profiles. Here comes my pitch to all of you. Let's think about n is equal to 1. I was never a fan of big studies. I couldn't understand from my childhood days of like residency and fellowship of how, in all PCOS women, or let's take a specific group of all lean PCOS women, this is what we are supposed to do, right? Because the lean PCOS women, from one patient to another patient, including their ethnic background, including what they do for work, is different, right? So what I'm suggesting to you is to think about n is equal to 1. What that means is that we are all unique, different individuals, and we are constantly changing, we are dynamic. Like, how can we cater to the success of each patient, one patient at a time? I understand that there are big problems in doing that, right? You have to collect data, and you have to do studies, and you have to like have it scalable, right? That's the buzzword right now. You have to high throughput systems. We need to get these patients done. You can't have time for each one of these approaches. But the thing is, at the end of it, it's n is equal to 1. Each one of you in this room, if we were giving you one exposure, so let's change the temperature of the room here, right? Let's make it 65. Each one of you is going to have your own little nuance of how you like it or not, and how your body responds to it or not. And it can also depend on whether or not you were born in a cold country. So I was born in India. I cannot tolerate a cold draft. And that's not just like, okay, my body is different than yours. It's also my conditioning. Over the years and years, I just adapted myself, including the conditioning that came from my parents, to adapt to that temperature, right? So n is equal to 1 is the paradigm that all of the big data is using. It's already being used. So when I get the pushback of like, oh, this is not scalable, it can't be done for you, what are you talking about? Healthcare doesn't work this way. We need studies and we need evidence. It's being done already. And where do I see it every day? In social media. My LinkedIn, I bet you if we open the LinkedIn together right now, my LinkedIn is different than your LinkedIn. Because LinkedIn has learned what I like. So what we are doing is we are using big data, and it has revolutionized a lot of things in this world, including how we manage traffics, right? All the information about traffic, all of the geopolitical stuff is happening through big data. And with AI now, it's happening in healthcare. A lot of the companies that we'll be talking after our segment are talking about how can we make a lot of variables go through a system and cater to an individualized approach. So big data has revolutionized the social media algorithm. They are tailoring content to individual preferences. You know, this personalized approach is known as n is equal to 1. It caters to unique needs and interests of each user. There is vast amount of data that's going into these algorithms, and then preferences, behaviors, and interactions are coming out. And basically, there is enhanced user engagement and satisfaction. So the system is dynamic in social media. Like if I talk to somebody, my family lives in India, right? If I talk to them about tulips this morning, by the afternoon, somebody would be selling me tulips this afternoon. Have you guys ever noticed it, right? So why can't we do it for patients? So what that means is, can we use big data analytics to analyze comprehensive patient data to enhance outcomes and experiences? Basically taking information from their genetics, their lifestyle, medical history, environmental influences, and what they like and don't like. Take all of that and kind of automate that process through the algorithms and with the large learning models. Like my ChatGPT has now learned the language that it wants to talk about genetics, right? So we can take all of those information and tailor to each patient's unique profile and optimize and increase success of the patients. So what I'm suggesting in this segment of my presentation is, can we take a lot of the information upfront from the patient, put them into algorithms, and then at the end of it, what we are now doing is losing that human touch. The physician is so busy taking care of the other things, and the medical team is so busy taking care of all that has to be done that the human connection of the patient with another human is changing. So taking the big data and using it in algorithms, making the plan individualized for that patient, and then using the humans that are available for that patient's interaction and make their journey a little bit easier. So this is what I'm suggesting. We take unique genetic and genomic information. My biggest pet peeve is finding out after 3 failed IVF cycles that they had a balanced translocation. Just makes sense? Like, we have to help patients through an algorithm that's cost effective, that's, is in a way going to take the high risk patients out in the broad sense. Those who are mosaic Turner patients should be already knowing that they're mosaic Turner and not have to go through an IVF retrieval or 2 for us to find out. So like the genetic information, the one written on the DNA, let's take care of that in some way. And then we also take the genomic information, like what are they currently doing and what is their different environmental risk factors. What ethnicity are they? How do they metabolize drugs, right? Are they going to tolerate the FSH or not? What is their age, BMI, stress levels, and then what is the male factor? And male factor in itself is a segue of like how big that is. Like right now, our assumption that we're gonna take this sperm sample and inject the sperm into the egg and we're gonna get a beautiful blast is not the right approach for it to be. The sperm is not just bringing the genetic information, not those 23 chromosomes, it's bringing these microRNAs that can change from time to time, right? So focusing on that, and then toxins. So if your patient is doing an amazing job with her diet and lifestyle and everything that you said, and she's not stressing out, but then every morning, this poor lady who doesn't know, she makes her smoothie in a jar that has BPA or phthalates, it's a done deal. You're gonna get some euploid blast, we are going to get away and you're gonna get a baby out of it, but we could have done better if we knew exactly what she's doing in all of her things. And this changes from patient to patient. So then we do an individualized ovarian stimulation, and Rohey Horowitz is gonna talk about like how can we use AI in like making these personalized protocols, and then individualized embryology lab approach. Sometimes we have to transfer a day 3 embryo. There's just not gonna be blast in this couple. They just don't have the right information for the genomic activation to happen outside in the lab on day 3 to make a blast in their situation. And then I was talking to somebody, we had a really nice conversation. They had a child from a transfer of a morula on day 5. The embryo was slow. It was not ready to be transferred. So if we had not transferred, and they have a beautiful baby child now, right? So it's like individualized embryology lab approach. The embryologists know what they are seeing. They have a developmental stage of the whole process, and then that will lead to a successful IVF. I think Dr. Cooper already covered it, but like these are some of the things that we are doing clinically. So we are incorporating the big things, but like in a PCOS patient, metformin has a big role to play in changing the epigenetics of what is happening in that individual, catering their stimulation protocol a certain way. The same thing with like endometriosis. You could do all you want in the lab with your amazing techniques. If they have inflammation going on actively in an endometriosis patient, their egg quality is going to be worse. And in the same regard, if we change their inflammation 3 or 4 or 5 months, and today I heard that cohort is starting 6 to 8 months ago, like if we started that time ago and endometriosis is better controlled, we are going to have a better cycle. We are going to need less number of cycles to get there. And then for DOR, we have to dose medications a certain way. If they have differences in like FSH receptors, Gonalef is not going to work and we have to have a different medication. So I'll leave you with that, and I hope that I've given you some ideas of what is our next frontier. I think it's like N of one, changing, solving problems, and kind of bringing everything together to serve that one patient right in front of you. Thank you so much.